Okay. So next up, we'll have Annaliese Beery talking to us about selectivity and reward in Vole relationships.
There we go. I think I got it. Can you hear me okay? Great. Oh no, don't go away. Oh no. This was so funky. You touched it. Great, excellent. So this is actually my third Vole meeting, and I'm really excited to be here, and I'm really excited to hear that there's another one coming in 3 years. So thank you to the organizers. Today I'm going to talk about some recent work that we've been doing in my lab on operant conditioning and social reward in voles. But I always like to start with the big picture. So just to kick things off, the broader question that we're focusing on in my lab is why would an organism be social, and what accounts for some of the variation in sociability and sociality? So of course, across the animal kingdom, there's tremendous variation in social behavior, all the way from this tree shrew over here. For a mammal, they exhibit very low social behavior, all the way to highly social species. These are bats and gelatinous. So group living actually provides a really nice marker for the kinds of social behavior that we're interested in, and provides a jumping off point for asking the comparative question, what causes some species or individuals within a species to be more social than others? Of course, whenever you ask this kind of why question, you can do it in different ways, and I just want to make a nod to the fact that there's lots of really great evolutionary or behavioral ecology research on why one might evolve to be in a social group. But then as neuroscientists, we often ask why from this sort of proximate or individual level perspective. So from that individual that's going to engage in social interactions, what's actually going to either motivate it to be social, oh yeah, so those things contribute to survival and reproduction. But from the individual perspective, what actually causes social motivation? These are a bunch of teenagers in a group sit. Boy, does that require social motivation. Or in instances where you're hanging out in a social group, but it's maybe not about something so pro-social, how do you put up with other individuals? So this is a scene from a subway. Of course, when we talk about social behavior, we all sort of talk about it very generally, but there are lots of different kinds of social behavior. I would joke that I could have put voles up for all three of these pictures. You can study parental behavior, maternal behavior, paternal behavior, alloparental behavior, social monogamy. And these two sorts of relationships on the left are really the best characterized and best studied, at least within mammals. And I consider both of these to be fundamentally reproductive relationships that are probably strongly reinforced because of that. And then over here on the right, I have to kind of throw out a new word, and I use the word peer relationships to describe these relationships between individuals that are not reproductive partners and they're not parent offspring, but they're members of social groups. And that's what we really focus on and ask whether it's fundamentally similar to or different from other forms of social behavior. And then you can say, what species should we study it in? Well, how about voles? Actually, we like to take a comparative perspective and study this in lots of different species. These are some of the models that we work on in our lab, both through collaboration and directly within our lab. But for today, it's just voles. All right, so I don't need to introduce prairie voles to any of you, and I probably don't need to introduce meadow voles much, but I will say that not only are they used as a contrast to meadow voles because in the wild they're not socially monogamous and they don't have biparental care, but what we use them for particularly is that they exhibit seasonal group living. I think that's okay. I'll wave my arms. They exhibit seasonal group living, and I stole this picture. It accompanied an article on seasonal changes in the microbiome of meadow voles as they overwinter. But their group huddling is one of the main characteristics that we use them for. And just to show you some of the original field data that we get this from, so these are data from Dale Madison, radio telemetry data defining the home range territories of, in this case, several female meadow voles. So each of these polygons represents a female vole. That's what it looks like if you go look at it in July. If you come back a month later in August, remarkably, those female territories are pretty much in the same place. So they're well-defined female territories. But if you come back in the winter time, meadow voles don't hibernate. And so in order to make it through the winter, they have to change up their solitary act. And in fact, their home ranges collapse and they huddle in groups. Okay, so this is controlled or moderated by several environmental factors in the field: temperature, plant compounds. But the main one, and the most predictable one for any seasonal mammal, is the length of the day. You can't have a short day in the middle of the summer like you can have a cold day in the middle of the summer. I recently put together a review, I think it came out in January, looking at what do we know about meadow voles in terms of what regulates their seasonal changes in social behavior as they go from solitary and territorial to group living and affiliative. And I'll divide these into sort of behavioral changes and physiological changes. There were behavioral changes in the field that are well described, behavioral changes in the lab with photoperiod, and physiological changes. And I'm not going to spend any time talking about these today, except to highlight that there's a wide variety of behavioral changes that all go along together. So everything in who they like to sniff, who they like to huddle with, how tolerant are they of strangers, how anxious are they, and many of these go along with changes in the brain that in some cases, when you manipulate these systems, are causally related to changes in social behavior. All right, well, let's get to that huddling with familiar partners. So we like to use voles to study selective social relationships. And in this case, when I'm talking about the selective social relationships, I'm usually talking about relationships between peers or same-sex individuals. So same-sex huddling preferences have been demonstrated in both meadow and prairie voles. And here I'm showing you some data from meadow voles housed in short days. That's the day length where they're affiliated with each other, not long days. And prairie voles, it turns out it doesn't matter what day length you house them in, and that was work done by Nikki Lee, my graduate student, showing that in long days and short days, prairie voles show same-sex partner preferences for familiar individuals. And we sort of wondered, well, everybody talks about selective preferences in voles, and we know that monogamy is really rare in animals, but how rare is this familiarity preference for peers? Nobody tests mice or rats or the other sort of semi-social rodents in the same way that we test prairie voles. We test them in 10-minute or 20-minute tests, and they're under cups. They're not tethered where you can have them interact freely. So a couple years ago, we decided to throw mice into partner preference tests and voles into the test that we normally use for mice and see how they compared to each other. And lo and behold, mice do not exhibit familiarity preferences. In fact, they don't even really like each other in these tests. They don't huddle a lot, and they spend most of their time in that central unoccupied chamber. Oh, well, what about rats, we thought? So we tried it with rats, and it turns out that rats also don't show familiarity preferences in a partner preference test. Now remember, this is all same-sex. And then we thought, well, what about a really social animal like a daegu? So we collaborated with Nathan Insel's lab. He has dagus in his lab. And we ran partner preference tests in dagus, which look really cute in those collars. And the dagus spent a lot of time huddling. But again, they didn't have familiarity preferences, so they could distinguish. We can tell from detailed analysis of their behavior that they can tell partner from stranger. But individual dagus would actually go back and forth and huddle with both the familiar and the unfamiliar animal, which is fairly consistent with their behavior in the field because they have flexible mixed-sex groups that are changing over time. So they seem to have a high level of social interest even in unfamiliar animals. All right, so voles are weird in their selectivity. That's kind of good for those who study voles to say, look, this is a really useful organism. If you want to understand that social selectivity, where does that selectivity come from? Does it come from motivation towards familiar animals? Does it come towards lesser fear of familiar animals or more tolerance of familiar animals? And so the first thing we wanted to look at is just how motivating are different types of relationships. For this work, which we've been doing for 3 or 4 years now, which is kind of amazing how time flies, but we did this work in prairie vole mates and peers in order to look at the difference between peer relationships and mate relationships, and whether or not those mate relationships are in fact more highly rewarded than peer relationships. And then we also looked at peer relationships between prairie voles and meadow voles to say, hey, are these relationships mediated more similarly across types of relationships or across species? So is it more, are prairie vole peers more similar to prairie vole mates or more similar to meadow vole peers? And of course, in both of these contexts, we're looking at both familiar and unfamiliar conspecifics. All right, so how do we measure that reward and motivation? not with a partner preference test. So the partner preference test has no barrier to huddling. It's 3 hours long. For all we knew before testing mice and rats, the voles huddled because they got bored. Well, the mice and the rats didn't huddle, so it's not boredom alone. But there's no way to assess reward in here, so what we did was we just first started by trying to make it harder for them to get over there. And we tried putting contact paper on the floor, and we put spiky horsehair brushes on the floor, and we made mazes, and we put trays of water. And what we learned is that voles love obstacles. They think it's environmental enrichment to climb on those little spikes, but they won't cross the water, period. So, fortunately, there are some established methods for assessing social reward, and we had much more luck with those. So the first of those that we used was the condition place preference assay. And the social version of this assay was developed by Jakob Panksepp and has been used in a number of different studies to look at the social reward associated with young pups or social reward dependence on oxytocin. And the fundamental way you run this test is essentially you take some cue, in this case a novel bedding that the animals have never been exposed to before. And in this version of the test, we used 2 equally preferable beddings that animals had never been exposed to before. We gave them 6 days of alternating cohabitation on one randomly assigned bedding that was the social bedding, and then they'd go to the isolated bedding, the other one, then they'd go back to social, back to isolated. After 6 days of alternation, you give them a post-test and just see have their preferences changed. Okay? We did this in meadow voles, in peers. We did this in short days and long days. We did it in prairie voles, in short days, because we didn't know yet that they still like each other in long days. And we did it in mate pairs. And all of these were long-term partnerships where animals had been together for at least a month. And the photos at the bottom, this work was primarily done by Nastasha Goodwin in my lab. So what we found was that, maybe not surprisingly, we found conditioning in the mate pairs, but we only found conditioning in the mate pairs. So only the mate pairs formed an increased preference for the socially associated bedding relative to the solitary bedding over time. And that was true in males and females, and you can see it from the pre to the post-test data, and you can also see it just in the time between groups, the time on the CS Plus data after testing. So what we can say here is that peer-associated cues are not as strongly reinforcing as mate-associated cues. Now there's another version of this test that gives you a little more headroom to find conditioning, right? If an animal happens to prefer the socially associated bedding in advance, they don't have that much room to increase their preference for that bedding. So you can also bias the animals by making sure that using a yucky bedding and making the yucky bedding get associated with social cohabitation. And when we do the test that way, what we find is that even under that circumstance, the prairie voles became borderline, but the meadow voles still didn't condition towards pure social cues, and in fact, they conditioned away from them. So that really is starting to fit in with the sense of social tolerance being more what's changing in the meadow voles seasonally than a sort of a pro-social reward process. And I'll also say that you should come to Nikki's poster tonight, that it turns out that when we use new partnerships instead of pairs that have been together for a long time, we actually do see social conditioning in prairie vole peer pairs. And she can tell you more about that. Nikki can also tell you more about the role of dopamine. So we've done some studies looking at the role of dopamine signaling in peer relationships in particular. We already know from work done in this room that dopamine neurotransmission is important for the formation of opposite-sex partner preferences in prairie voles. And the same is not true for same-sex relationships in meadow voles. And so what Nikki has done is manipulate dopamine receptors pharmacologically to say, hey, well, what's the effect on prairie vole peers? Is it more like the meadow vole peers, or is it more like the prairie vole mates? And that's work that you can see at her poster as well. All right, but the gold standard for measuring motivation has long been operant conditioning. It's certainly the most quantifiable. It's also the hardest to do. And it hasn't been used very often for social reward, but it has been in a few contexts. So some of the older studies that you may have heard of, you can use social rewards getting males to bar press for access to an estrus female, where you drop an estrus female into the cage. Alison Fleming's lab showed that moms will bar press for access to pups. And when you're like, how does that actually work? This is one of my favorite quotes from a paper. With each bar press response, a rat pup, rather than a Fruit Loop, was delivered down a gentle chute into the hopper. So we don't do it that way, but you can. And then more recently, it's been shown that actually social reward by aggression works in operant conditioning settings. So animals will bar press in order to get access to an animal to go beat up on. And then there have been a handful of other studies. Elliot Albers' lab has recently come out with some work using operant conditioning, but a different task. Instead of contact paper or spiky hairbrushes, they use a weighted door and measure how animals will cross through that weighted door to get to other hamsters. Lisa here, who's here, did her undergraduate work in Tim Hackenberg's lab on social reward with operant conditioning in rats. And there is one Vole paper that proves that voles can press levers in order to do operant conditioning. It was done in long-day voles and only with strangers, but it gave us hope that we would be able to train our voles. So this is work that was done by Sarah Lopez, primarily in the lab, although it's spread beyond that. And I first wanted to show you the chambers that we use. So on the left we have, this is a food reward chamber. Food is gently delivered down a chute to the animals when they press a lever, and this is how it works. All right, so go get a pellet, go get another pellet. That animal's on an FR1 schedule, a fixed ratio schedule, where every time they press the lever, they're going to get a pellet. So we train animals on this schedule while they're getting used to what the lever does and how it works, and then we move them on to what's called a progressive ratio schedule, where they have to press more and more times to get subsequent rewards. Okay, then we move them on to this apparatus, the social reward apparatus, and in this case, now we have a motorized door that's activated by the lever. So the door goes up, the animal crosses over into the other chamber, or at least has the option to. And then here's a little video from that. This video is actually from a later apparatus. So you can get the idea. Press the lever, the door goes up, go hang out. And I was telling someone earlier that the noise of the door becomes a really salient signal for them, so that as they start to hear the motor of the door, normally you have to shoo them back after they have that minute of social interaction, and then they press the lever to go get more social interaction. But some of them over time hear the motorized door start to drop and run back over to avoid your hand, and then they'll press it again to go back over there. So smart voles. All right, so here's the timeline we used. We thought it was really important. So first we start with shaping and training them, getting them used to pressing the lever, getting them to understand that the lever is associated with the reward. And then we wanted to quantify their pressing for a food reward. And we felt this was really important in part because that one prior vole paper had a lot of voles that never pressed. And you never know, did they not press because they didn't learn, or did they not press because they weren't motivated? So we wanted to make sure that all of our voles were regular pressers for food rewards, and we knew how much they pressed for food. We then turned them over to a social stimulus and on a fixed ratio schedule. And then once they understood how the apparatus worked, we did PR1 testing for 8 days with the partner, tethered in the social chamber. And then we did 8 days with the stranger, tethered in the social chamber. And half of them got the partner first, and half of them got the stranger first. Okay? And after that, we went on to do a series of controls and extinction. We tested them with empty chambers. We tested to see what do they do when the lever doesn't do anything. All in all, each animal had over 50 days of active hands-on testing. And we could run about 6 at a time in that kind of labor-intensive way. And once you see the number of groups that we had, you'll understand why this experiment took a couple years. All right, so we tested a whole bunch of bizarre codes that don't make any sense. So let me sort these into groups. We've got male, female, Microtus ochregaster, and female, male. So we've got animals in opposite sex pairings. We've got male, male, and female, female. So males pressing for males and females pressing for females. That's our prairie vole peer group. And then we've got meadow vole peers, females pressing for females. So in the meadow voles, it's only the females that have this seasonal change in their social grouping behavior that's really dramatic in their social behavior. So we just tested the female peer pairs there. All right, so first I just want to show you the data from some controls. So good news, it turns out that there weren't any dramatic species differences in just how much they were willing to press for food. We kind of thought, well, what if prairie voles just learn the task better and are better pressers or something like that? But all the groups pressed the same on average, males, females, prairie voles, meadow voles, for a food reward. We then looked to see, well, are there individual differences? Do we have super pressers who just press a lot and individuals who don't press very much? And so we looked at their response rate, or how many responses per minute of testing they gave for a food reward, and then how much they gave for a social reward. And anytime the lever is up, or the door is up, the lever is inactive. So we scale it by the number of times the door is raised. And what we see here is that there's absolutely no relationship between how much they press for the food and the social reward. So we interpret this as either lever pressing is reward specific, or at the very least it's not individual specific. Okay? Not shown here, but also true, pressing extinguishes in about 5 days. In the absence of a social reward, it drops off very nicely. And then voles will press for access to an empty chamber. So when they're pressing to get access to that social chamber, there's two components. One is I want to go over there, and the other is I want to be with that animal. And so that's something that we take into account. Okay, so first I'm going to show you the species differences. And these nicely parallel the condition place preference results that we got looking at prairie versus meadow voles. So these are females pressing for other females. So these are prairie vole, I forgot the pointer doesn't work. Prairie vole females on the left pressing for their partner, and then in the lighter tan bar, that's pressing for the stranger. And the same animal gets 8 days of pressing for the partner, that's one of those dots, and 8 days of pressing for the stranger, that's the other one. So they're connected because all the animals did both. And so you can see they press significantly more for their partner than their stranger. That's also true in the meadow voles. They press more for the partner than the stranger, but they press less overall. There's a species difference in total pressing. And if I were to cram the control behavior on the same graph, what you'd find is, here's how much they press for an empty chamber. So they prefer the partner to the stranger, but they're not actually pressing more than they would for the empty chamber, which the prairie voles are. All right, so prairie voles appear more motivated by social interaction than the meadow voles. The second comparison we wanted to do is within types of pairings in males and females in prairie voles. So here we have females pressing for males, partner and stranger, and females pressing for females. And in both cases, you can see that there's a significant effect of familiarity. They press more for the partner than for the stranger. And actually, that was true whether they're pressing for opposite sex or same-sex partners. Okay? So the conclusion for the females is females are more socially rewarded by their partners. Totally consistent with what we would expect from partner preference tests. Here are the male data. You don't get the same thing. So males press a lot for females, and they press equally for a familiar and an unfamiliar female, and they press less for males, and they press equally for a familiar and an unfamiliar male. So here there is absolutely no effect of familiarity, and there is a strong effect of whether they're pressing for a male or a female. So the males worked hard to access any females. That's how much pressing they did. But once they press and the door goes up, they get to go and interact with that other animal, and we can look at the behavior they exhibit while they're there. So how much do they actually huddle with the animal when they get over there? Now things look exactly the same between the males and the females. So here we have huddling out of the total time that that door is up. And you can see that whether they're in an opposite sex or a same-sex pairing, whether they're males or whether they're females, everyone prefers to huddle with a partner. Now, here's a catch. They don't have a choice between a partner and a stranger here. So when I say they prefer to huddle with a partner, what that means is when the door is up and it's a partner over there, they huddle. When the door is up and it's a stranger over there, they go in, but they don't huddle. They do other things. So social reward and selectivity seem to be different. They really only selectively huddle with the partner, but they seem, at least in the males, they seem to be rewarded by strangers. So why so much interest in strangers by the males? Well, after we got that somewhat surprising to us result, we went back and rescored 320 videos to figure out what were the males actually doing when they got into those chambers, or what were all the animals doing in those chambers. And two things that we were particularly interested in. One was how much mounting or sexual behavior did they display? So strangers represent mating opportunities, so do partners, but strangers represent novel mating opportunities. And what we found was that 4 out of the 8 males who were in male-female pairings mounted strangers when they had the opportunity to, and no animals in any other groups did. So males never mounted their mated partners. Then we also looked at at whether strangers were representing opportunities for aggression. Remember that mice will bar press for access to another mouse to beat up on it if they're a previous winner. And we found that there was more aggression towards strangers, not surprisingly, in both sexes actually. So aggression was a little bit higher in males, not a whole lot, and it's always higher towards the strangers than towards the partners. So one thing that may help us to understand this difference in males a little bit better also is to say, well, what would happen if they had access to both a familiar and an unfamiliar female at the same time, right? Maybe they're only going in and interacting with that unfamiliar female because it's the only choice, right? So here we decided, well, what if there's a choice between the partner and the stranger? And so that's where we have been going recently. We've been doing experiments in a social choice apparatus where we can put the partner and the stranger on opposite sides. The voles are actually capable of using 2 levers and going to better and worse chambers. I think 2 is probably their limit, but I'm not totally sure. We've been doing some work adding pharmacological manipulations onto the operant paradigm. And then recently, for about the last 9 months, we've been collaborating with Zoe Donaldson's lab, looking at social motivation and partner loss, and we're hoping to do some more collaborations related to the operant conditioning setup that they're automating in their lab. And then, in honor of having had Ben Dancer give the talk a couple talks ago, I just also wanted to say, and we're super excited about RFID stuff as well. So you might remember that the title of my talk said Life in Groups, but almost all social behavior is studied in the lab in dyadic interactions, or sometimes we'll have 2 stimulus animals and 1 focal animal at the same time. And one of the things we'd really like to be able to do as we try to study and understand group living is to study and understand animals that are housed in groups. And so one of the difficulties with this is always that voles are hard to tell apart, they're hard to mark in a permanent way, and so RFID systems will allow us to house and track in an automated way animals living in groups. So with that, I want to acknowledge only a few of the collaborators are listed up here. One's related to some of the work today, but a great many people who make this work possible.
Let's open up for questions. Any training? Go ahead.
Awesome, super cool stuff, obviously. With the operant conditioning, because I've also gotten a couple questions about how to account for the empty chamber kind of reward level. I'm kind of curious how you disentangle, so the fact that an animal will press to go to an empty chamber makes sense, right? But do you really think that the motivation to open the door to go to a chamber that has your partner in it, or another animal, is like a linear combination of the motivation to see your partner and the motivation to enter an empty chamber? Or is it that there's something, there's a more salient feature now that your partner's there, and that's really what's driving your motivation, and you've forgotten all about the fact that, oh, I'm also going into another door or something.
Yeah.
Right.
I don't know if I have a way to answer whether it's a linear combination or not. I think it's informative to us when they don't press more for a socially occupied chamber than an empty chamber. Then we can say, yeah, that doesn't look like social motivation. It looks like, I hate that chamber less than I like that chamber less than I like this chamber. The one that's occupied by a stranger is less appealing to me than the one that's occupied by a partner. So I don't know that it's linear, but I do think it's something we have to account for. It's just how much do they, because they like to explore and they like to make the lever do something, right? This is why extinguishing isn't immediate. They're like, well, maybe it'll still work. Yeah. Yeah.
Have you tried to give them an option between an empty chamber and a chamber with a social?
We haven't done empty versus social, but in our 2-chamber setup, we have done what we call the good-bad test, which is really good neutral, where we make one chamber have fluffy bedding and Cheerios and good things, and one of them be empty. And then more recently, I made a little 3D printed water tray that fits in there, so it's actually a good-bad setup, just to show that they can tell the difference, because we initially started doing this comparative social work in the males, and we expected them not necessarily to have a preference for the males versus the females, and most of the males didn't prefer, the familiar over the unfamiliar, And then we realized, we need to know whether they can actually tell the difference. Yeah, so.
So I have actually two questions, but they should be pretty quick. The first one is, so for these peer relationships, were these like, how long were they established? Was it like since weaning, or were they like taken from their weaning group and then paired with a new animal for a short period?
That answer differs by study, but what we've shown is that you can either house animals together from weaning, or you can pair them in adulthood. They'll form equally strong preferences. In the socially conditioned place preference paradigm, you get slightly stronger conditioning when the preferences are relatively new, but even then they're not new. Like, it takes 24 hours to form that peer preference in adulthood with a new animal. But in the socially conditioned place preference paradigm, then you house them in alternation over 6 days, so they kind of get 3 additional days of housing with that peer. So it's sort of new in that grand scheme of things. But it takes them 24 hours to form a preference for a new social peer after they've had a prior housing, or you could do it where they've been housed together all along. Yeah, and we do it both ways, depending.
And then the other one was that, so the operant conditioning, it was like 8 days per, like they got 8 days of a stranger, right, and then 8 days of a partner, or vice versa.
Yeah.
So for the stranger, I assume it was a different one every time, but did the response towards like over time, did they get used to the idea of this is an animal I'm not gonna know? Or like did you observe any
I think
change
so.
in time?
So this was the very first study that we did, and we wanted to play it safe because we didn't know how long is it going to take for them to really learn the task, and we thought, well, maybe we'll run it for 8 days and use the last 4. So we looked at the data over time, and it was actually very consistent. So we analyzed the last 4 days of data versus the whole 8 days of data. It seems to be pretty similar. We've done some studies where we manipulate pharmacologically, where we give the animals a drug and then we test their response rate, and we've switched them after every 2 days, and they seem to get confused. So they do seem to learn what paradigm they're in and know what to expect, but we didn't get a big change in behavior over the 8 days. You do get a bit of an effect. The ones that did the partner first before the stranger have overall higher pressing levels than the ones that pressed first for the stranger and then for the partner. You can see that here are their stranger days, here are their 8 partner days, but they have an overall lower mean than the ones that pressed first for the partner and then for the stranger.
Have you tried some kind of progressive ratio schedule for partner versus stranger?
What schedule?
To look at the breakpoints, progressive ratio
Yes.
schedule,
Yes.
where you ask the animal to work harder and harder
Yeah.
to get through more.
So all of that work that I just showed you, all of that was, I happened to show you lever presses, but I could have plotted the breakpoint. So that was all on a progressive ratio schedule. And, I go back and forth about do we do the breakpoint and the number of lever presses, but they're completely redundant pieces of information. You get a little bit more from the total presses because if they don't quite make it to that next number, but they show you the same thing. So we plotted both sets of data. Breakpoints give you exactly the same answer, yeah.
Annaliese, thank you. That was really interesting. I'm curious to know if you think that the mechanisms that are responsible for this social reward preference are redundant with or separable from that which underlies motivation, social motivation. Yeah, I'm wondering about preference versus motivation, because what you have here is the ability to measure the motivation, whereas the partner preference test, they're both kind of muddled together.
Yeah. So, I think that's what we're trying to separate here is how motivated are they, how much will they work to get to that other individual. And I think from these data, what I would say is that in the partner preference test, you're sort of seeing, as you said, both of those things together. You're seeing some social motivation, but also you're just seeing the relative desire to huddle with a known versus an unknown animal. So I think this is actually more pointing out important differences between what you measure in the partner preference test and motivation. And I think the partner preference test also combines tolerance and motivation completely. So the meadow voles, for example, look exactly like prairie voles in same-sex partner preference tests. They strongly prefer to huddle with familiar individuals, and they huddle quite a lot. But when you ask them to work to get to the partner, or when you ask them, do you like stimuli that are associated with being socially housed, they seem less motivated than the prairie voles.
Okay, we'll just take this one last question.
You mentioned that when you put your hand in there to shoot them back into the original chamber, that's like an aversive stimulus for them. Does that change their behavior on subsequent trials? How many trials can you do in a session, things like that?
Yeah, so I mean, we've had one or two bulls learn that they can just shoot themselves back in, but they get really habituated to us when we do the operant conditioning. So everybody knows bulls are nasty, they're no fun to work with. These bulls are the friendliest bulls you work with because you handle them every single day, 50 days of testing, in and out and shooing them back and forth. And they go in for a minute, and then you take them out, and if you can't shoo them across, you actually lower the door, pick them up, put them in the center, and they just press and go right back into the social chamber. So it can't be too aversive, because they really are continuing to press and go over there. They really do seem interested in going over the other chamber. And that was also part of the reason why we do such extensive periods of testing. They really have a chance to get used to how the study works. So if it's aversive, it's not so aversive that they're avoiding it. All right, thank you all.